HPL seminar. Today we'll be hearing from Fabian Hoitz. Fabian will be defending his PhD in biomedical engineering next week, and the title of his dissertation is Layer-Wise Relevance Propagation in Biomechanics. His PhD was advised by Dr. Ben Onig and Dr. Art Kuo. Fabian completed his Bachelor of Science in Sports and Computer Science at Technical University of Darmstadt in Germany. Fabian then completed his Master's in Performance Analysis of Sports at Otto von Guericke University, also in Germany. His master's work focused on the preferred movement path paradigm and changes in muscle activity. Some of his proudest accomplishments include winning a MyTax Accelerate grant, receiving an Alberta Innovates Scholarship, and getting emails from other researchers in the field who have read and admire his work. Later this year, Fabian will transition to an industry role at Garmin. Some fun facts about Fabian is he was a shepherd for a year between his bachelor's and master's in Norway. Another fun fact is he studied at 3 different universities during his master's. Finally, for a gap year during high school, he lived in southern Brazil. So I'll first turn it over to Fabian for his presentation, and then we'll have a question and answer period to follow.
Thanks, Emily. Does this come through?
Looks great.
Awesome. Okay, so biomechanics has entered an era of big data, meaning that the data that we collect and analyze these days has grown tremendously over the recent years. Another thing that comes with this is that the data sets that we have are now way more complex and comprised of more and more variables than they have been ever before. Let me kind of try to illustrate that on an easy example. Say we are interested in a relatively standard biomechanical question, such as the difference between 2 shoe constructions. To answer this question or to investigate this question, we would invite a couple of participants into the laboratory, have them wear the 2 shoe constructions and run for us while we collect some data. Now, the data that we want to collect could include joint angles here on the left using motion capture. It could include movement vectors collected via IMUs. We could investigate plantar pressure, ground reaction forces, and muscle activity. Now, naturally, we would normalize the data modalities that we collect to stance phase. So we would express every different data modality here in 100 values, describing the moment of heel strike to the moment of toe off. If we do that, and if we also collect more than just 1 joint angle, say 3, for instance, and we collect IMU data from 2 locations and muscle activity from 2 or more locations, then a simple data set such as this would already be comprised of more than 12,000 variables. So this is quite the drastic number, and our traditional analysis methods, such as the novice or t-tests, usually become somewhat ineffective if we have to apply them over and over again to figure out what variables best show us the difference between the 2 shoe constructions. So what the field has done to adapt to this is moving towards more data science heavy methods, such as artificial neural networks, for instance. Now, artificial neural networks are great because we can essentially take all this data that we've collected, like this entire set of 12,000 or more variables. We can kind of train an artificial neural network model with this, and the model learns to predict how to best classify an unseen data set as either shoe A or shoe B. So in other words, the model itself has an internal representation of what the data for shoe A or the data for shoe B looks like. So we have some sort of a difference inside this model. Now, this is great, but the biggest problem here is that these models often kind of act as black boxes. So even though the model can separate these data sets from one another, and the model knows what's different, we as researchers are often left behind and have no real idea of what to look for and what the model used. So over these last 4 years where I've kind of worked on my PhD, I started to look into this problem, and I tried to find ways to kind of analyze these black box model and see if there's a way we can learn from them and see what these models actually find to be different in the data. And today I'm going to present basically the journey that I've been going through over these past 4 years. The method that I ended using is called layer-wise relevance propagation, and it was introduced by Bach and colleagues in 2015. Now, the idea of this method is relatively straightforward. Instead of starting at the beginning, so here on the left-hand side where we have usually the input represented, we would start on the right-hand side of the artificial neural network where we have the output. So for instance, a classification such as this data set belonged to shoe B. Knowing this output, we would then ask the question of the degree to which each individual parameter in this artificial neural network contributed to this output. So all these circles that we see here, these individual nodes, are somewhat a depiction of the internal parameters that this artificial neural network model has. And what we can learn with this approach, this analysis approach, is that, for instance, this node here at the bottom that is dark red would strongly influence this outcome. So it would strongly suggest whatever this node represents is responsible for this outcome. While this node at the top here, which is in a lighter red color, would not have a strong influence on this outcome. And we can propagate this information backwards through this model over and over again until we reach our leftmost layer, so our input layer, where we have the representation of the data that we understand. So we would essentially learn which data point that we provided the model with had a strong influence on our outcome and which data point had a weak or not a strong influence on our outcome. The studies that I'm going to talk about 3 studies today, and each of those studies kind of utilize this methodology and kind of try to explore how we can best use it in biomechanics. The first study that I'm going to present focused on kinematic and kinetic variables that we wanted to identify, that we wanted to use to identify novice runners. Now, all of us have this familiar feeling or had this familiar feeling before where we see someone walking at a distance and we realize, hey, I know this person, even though I haven't really seen their face before or today. And the question that we had was essentially, how can we figure out, or what variables can we look at to make this decision, to make this classification as I know this runner? And to answer this, we invited 50 novice runners to our laboratory. This was a relatively broad data set, so we had males and females in there, all of them relatively new to running, so a big variety. We had those runners run at 3.3 meters over ground, and we did this protocol on 2 different testing days that were 8 weeks apart. During these 8 weeks, we had 3 intervention groups. Now, this is a legacy component of the original purpose of this data. We kind of repurposed this data for this study because this was in the midst of COVID and I couldn't really collect my own data. So the intervention groups were a strength group. So these were individuals who received a strength training for the muscles surrounding the ankle joint. The second group was a balance training. So these individuals only did some light balance exercises. And the last group was a control group. So there was absolutely no prescribed training. As a result of this, we had essentially 20 steps per participant and per day that we could analyze. And the data, or these steps, let's say, were expressed through the data via motion analysis and Kistler. So we had joint angles and ground reaction forces that we would look at. The methodological approach for this study was relatively straightforward. We asked ourselves, can we design an artificial neural network where we present the network with data from these runners, so this joint angle trajectories and these ground reaction forces. And the network then had the task to essentially assign this data to one of the 50 runners that we had. So it was trained to match movement patterns to runners. Whenever this matching or this classification was correct, we would perform the layer-wise relevance propagation analysis, and we would learn what variables did the neural network use to classify this specific data set or this specific step pattern. So we would essentially have as many relevance patterns, i.e. patterns that describe the importance of the individual variables, as we had steps in the data set. And by averaging all these relevance patterns out, we would get an idea of what is generally important if we want to recognize a runner, if we have this data set available. Now, the graph that I'm showing here is essentially the combined results of this. And I know that the slide is somewhat loaded, so I will walk you through this step by step. First of all, on the left-hand side here, we have the respective movement trajectory that we used for this analysis. So we have the joint movements of the hip and the coronal, sagittal, and transverse plane. And then we have the joint movements of the knee, ankle, and the ground reaction forces, all in 3 dimensions. On the x-axis here, we see our normalized stance phase. So essentially the time period from heel strike to toe off, represented in 100 values, as I mentioned in the introduction. And if I now direct your attention to the very first row here, to the row that describes the joint trajectory of the hip and the coronal plane, then I can also explain the different colors. The colors here represent the importance of the respective variables for the neural network classification. So at the top here, we see darker colors representing a high relevance score. So these are aspects or movement aspects that were very important for a correct classification. Lighter colors, on the other hand, represent movement aspects that were not as important. If I now bring back the entire heat map here, we can see that there are certain trajectories, such as the sagittal plane of the hip, for instance, that on average were not as relevant to the neural network as, for instance, the coronal plane of the hip that we looked at previously. Another interesting aspect is that these black regions, so the regions that are of high relevance, are kind of spread out across this entire movement pattern. So we see some here in the early stance, we see some here in the later stances, and we also see them across all of the trajectories that we look at. So when we summarize these results, essentially, we learned that the most relevant variables for identifying novice runners were predominantly from the early stance. So if we would do a vertical summation, then we would see that the early stance is slightly more relevant than the mid to late stance. If we want to focus our attention on the different trajectories, then we learned that the up adduction and internal external rotation of the hip, the up adduction of the knee, the in eversion of the ankle, as well as the medial lateral ground reaction forces were very important. This study was kind of the first one that we've done to kind of explore this analysis method. And I think this is a great example to show how we can direct our focus onto the most important and most relevant aspects. Now, the next study I want to talk about took this analysis approach a step forward and tried to kind of build on this first study that we've done. So for this study, we tried to focus on kinematic variables only, and we wanted to identify elite runners, so a subset of runners. The idea here being is that hopefully different variables are important to classify within a smaller, more homogeneous subgroup than they were in the novice runners. For this study, we brought in about 20 participants. All of them were male triathletes that were training to finish the Ironman competition in under 2 hours and 30 minutes. These individuals ran a 4-kilometer outdoor run for us, which meant that we collected about 4,000 steps per participant, so quite some data to analyze. And the system that we used to kind of collect this data was the MVN link system. For those who don't know about this, it's an X sensor suit. So it's a full body suit comprised of 17 different IMUs that are located all over the body. So as a result of that, we would essentially have 18 kinematic trajectories that are coming from locations such as the spine or the upper extremities and the lower extremities. So we had full body kinematics. Similar to the very first study, we stuck to our strength and performed the same analysis. We would train an artificial neural network to match movement patterns to runners. Whenever a movement pattern was correctly matched, we would do the layer-wise relevance propagation analysis, and we would learn what variables were important to identify a runner, basically. And the next slide here also shows the averaged result again. Now, I know this is quite loaded again, so once again, I'll walk you through this. On the left, we have the joint angles. The first 6 here describe joint angles that were located around the spine. So L5S1, L4, L3 are all spinal joints. The next 6 joint locations here are joint locations describing the upper extremities. So we had shoulders, elbow, and wrist for the active and the passive side. I'm going to talk about this in just a second here. The last 6 joint location here were joint locations from the lower extremities. So we would look at the hip, knee, and ankle. Now we separate into active and passive side, basically saying that the active side is the side where the leg is currently in contact with the ground, while the passive side is the leg where that's in swinging motion at the beginning of this normalization period. Now on the x-axis here, we have the normalized values. So the 100 data points representing our gait cycle. Steps for this study were parsed slightly differently in that we parsed them from heel strike to the heel strike of the same leg again. So in other words, the first 60 values that we see on this graph represent the stance phase of the active leg, while the next 40 values, 60 to 100 here, represent the swing phase of the active leg. And if you can squint, sorry, I forgot to mention that, if you can squint, you see that each of these joint labels here is in the center of 3 rows, and these 3 rows correspond to the 3 movement directions of the respective joint. Now the color code is exactly the same, where we have brighter colors or white colors, meaning not important or not relevant, and darker colors here at the top, meaning very relevant. And I think the biggest takeaway from this slide is that all of a sudden, this relevance pattern or this average relevance pattern is not as clear anymore as it was in the first study. A possible interpretation could be because we have a more homogeneous subgroup and we have a lot more data, all of a sudden the network has to work a little bit harder. It has to look at a lot more variables. It has to consider a lot more variables to do a correct classification. Now, because it's so difficult to characterize the most important and the least important variables from this graph, we thought that we reduce the data a little bit and make it a bit clearer. So we essentially chose to isolate the 540 least relevant variables from this heat map, and we isolated the 540 most relevant variables from this heat map. Now, 540 variables expresses about 10% of the total data. And this number wasn't chosen arbitrarily. We did a site analysis where we looked at the performance of our artificial neural networks as a function of how many variables were included. And it turns out that these 540, so 10 to about 20% of variables, already give us classification accuracies better than 90%. So this is a very accurate model already if we limit ourselves to these variables. So the next couple graphs that I'm going to show here essentially characterize these variables in a bit more detail. On the y-axis here on the left, we see the percent or the count of variables expressed in percent out of these 540. On the x-axis here, we see the movement directions these variables describe. So we have adduction here in the first 2 graphs, rotation as the second 2 bars, and then flexion extension as the last bars. And the color coding, again here at the top right, represents whether or not these variables were from the higher end of the spectrum or the lower end of the spectrum. Now, I think that a relatively interesting aspect here is that flexion extension variables, so the 2 highest bars here, were derived from or were present in both the high relevance variables and the low relevance variables, like in the subsets. This would suggest that running in general is a relatively, flexion extension heavy movement. And we have information in those flexion extension movements that separates between our elite runners, but we also have information in these movements that is very generic, that does not separate between our runners. Interestingly, rotation movements seem to be a little bit more highly relevant. So it seems that rotational movements have more room for subject-specific movement patterns, while adductional movements seem to be a little bit low relevance. So these seem to be movements that are more generic across our investigated individuals. The next graph here characterizes our variables with respect to the step cycle. So on the x-axis now we have the 100% of our step cycle. Remember that about 60% here is toe off, and then 100% again is touchdown of the same leg. I think the interesting aspect in this graph here is that we have two very prominent peaks in the low relevance variable subset. So we have one peak at around here 10-ish%, and we have another peak at around 55%. So these peaks would correspond to our heel strike or our touchdown and our toe off within the data set. So it seems that all of the elite runners that we investigated have a relatively generic, so to speak, strategy when they are getting in ground contact and when they are pushing off the ground. Room for subject-specific strategy seems to be within these transitional phases where we have the transition from touchdown to toe off, or the transition from toe off to the touchdown again. So it seems that if we want to look at subject-specific characteristics, we might have to focus on the transitional phases, while if we want to look at more generic aspects where all of the elite runners seem to do something comparable, we have to focus on the touchdown and toe off regions of the step cycle. The third characterization here is now with respect to the joint location. So the first two bars here on the left represent joint locations around the spine. I tried to highlight that here in color on this little figure on the right, where all the yellow dots represent the spinal joints. The second in green here are the upper extremities. Now these are joints, like I said, shoulder, elbow, and wrist. I only show one side here, but the other side is included in this summary. And then the last bars are the lower extremities, so hip, knee, and ankle. And the interesting aspect here is that we see a similar picture as we did in the very first graph that I've shown, in that spinal joints seem to be, or variables contained to the spinal joint have high relevance, but also low relevance at the same time. So once again, the spinal motion has unique or characteristics that separate between our runners, and it has characteristics that do not separate between our runners, so rather generic characteristics. Luckily for us, lower extremity joint locations seem to have a high relevance, meaning that all this focus that we've placed in the past on lower extremity variables when we compared individuals and runners was well placed, because it seems that here unique strategies really seem to shine. The upper extremities, on the other hand, seem to not contain a lot of runner-specific information. Now, whenever I say that these variables separate well between runners, or they don't separate well between runners, I basically make an interpretation. And this is based on the fact that we trained this artificial neural network model to separate between our runners. But we wanted to do a little, let's say, sanity check to see if this is even a valid interpretation. So we wanted to visualize the interaction of the different runners, different movement patterns in these subsets of variables. We essentially performed a principal component analysis for each of these subsets that we selected. And that would mean that we would obtain a subspace of low relevance and a subspace of high relevance into which we could project our variables and see how they interact. We could project our movements patterns into these subspaces, because the principal component analysis is great for visualization. Now, this graph here shows exactly this outcome. And I know this slide is quite loaded again, so I will walk you through it. On the left here, we have the graph representing the low relevance subspace. So we have the lighter end of the spectrum. The individual axes here represent our first three principal axes. So these are, so to speak, the axes that represent the highest variability in our data set. On the right-hand side, we have the same depiction for the high relevance subspace. Now, the colored diamonds, squares, triangles, and circles we see here are 100 randomly selected step patterns of 4 participants that we projected onto these subspaces. And what you can see here on the right is that the high relevance variables seem to really separate the movement patterns of our 4 subjects here, 4 runners. While on the left-hand side, these movement patterns seem to overlap quite drastically. So there's really like not a lot of separation possible. While this is not an extensive analysis, it's a great visualization that shows us that the artificial neural network really did what we told it to do. It figured out which variables best separate between our runners. And this principal component analysis here, this visualization, shows us that we now can trust these layer-wise relevance or these relevance scores that we investigated previously in a little bit of a greater detail. Now, the last study that I want to talk about today is, it took a slightly different approach. One problem we often face in biomechanics is the effect of different footwear constructions on movement patterns. What we found so far is that these effects are often subject specific. So one subject might react in a completely different way to the same shoe than another subject might do. So we approached this problem from the perspective of artificial neural networks and layer-wise relevance propagation. And we wanted to see if this analysis approach can somehow help us with this problem of subject-specific footwear effects. For this study, we recruited 36 participants who came into the lab and ran in 4 different conditions. We had 1 barefoot condition, and then we had 3 shoe conditions that I illustrated here on the right-hand side. We had a minimalist shoe, or a barefoot shoe, whatever you want to call it, a shoe that is minimalistic in its construction. And then we had a very cushioned shoe, so one of a more conventional running shoe, let's say. The third shoe here at the bottom, in terms of its construction, sat somewhat in the middle between. It was less cushioned than our heavily cushioned shoe, but it was more cushioned than our minimalist shoe. The movement patterns that we looked at, or the movement patterns that we analyzed, were expressed through 2 kinematic trajectories, the ankle and the knee joint motion. And for each participant in each running condition, we collected about 10 steps that we could analyze. Now, the analysis approach for this data set was somewhat different in that we really wanted to focus on subject-specific or single subject models. So we essentially trained 36 different artificial neural networks, each specialized to the data of 1 subject. The goal for all of these networks, however, was to match the movement patterns of the 4 different running conditions to the correct running condition. Whenever this was done correctly, we would do the layer-wise relevance propagation analysis, and we would obtain a relevance pattern for this specific model, or for 1 specific model, that would tell us which variables were important when you wanted to look, let's say, at 1 shoe condition, or when you wanted to look at the barefoot shoe condition. So we would have a total of 144 relevance patterns that we could look at. The last step in this analysis was essentially to see if there are similarities across these 144 relevance patterns. And to do that, we used a cluster analysis. We used a self-organizing map to group relevance patterns according to their similarity. And we would do this 4 times, 1 time for each running condition that we had. And moving forward, I will only present the results for 1 of the running conditions, because otherwise it would kind of go too long. This presentation would kind of be too long. So this graph here shows the matched or the clustered relevance patterns of the individual participants that we have. On the y-axis, we have the participant IDs. So these are the actual participant numbers. And this vertical gap here represents a break in terms of, like, the first graph describes 1 group of similar relevance patterns. The second graph here describes a different group of similar relevance patterns. The x-axis here describes our movement trajectories that we looked at. So the first box is knee extension, the second box is knee adduction, the third knee internal rotation, and so on. And the color coding is very similar to the previous slides, where light or white represents low relevance and black or dark represents high relevance. And what we see from this very first picture here is that the first group of people, these about 10 individuals here, were all individuals who seem to have a high relevance score when looking at the ankle inversion here. all of these individuals seem to have, or the models trained on the data for these individuals seem to place a high emphasis on ankle inversion. Now, the second cluster here at the bottom also had some relevance on the ankle inversion, but it had, in addition to that, a lot of relevance placed on the ankle adduction. So that seemed to separate these two clusters. These are about half of the people that we had in our data set. So the second graph here describes the other half of people. So in total, we had 4 groups of people, or 4 groups of similar relevance patterns. The third cluster here at the top seemed to place a lot of relevance on the ankle adduction, but we seem to be missing this focus on ankle inversion that we saw in the previous 2 groups. So this seems to be the separating characteristic for cluster number 3. The graph here at the bottom represents the last cluster, which in my opinion just didn't fit into any of the previous groups. We do not have any, or we do not have a lot of relevance on the ankle inversion, and we do not have a lot of relevance on the ankle adduction. Instead, there seems to be some relevance placed here on the knee adduction, and then some relevance kind of scattered throughout the rest of the trajectories. Now, these patterns, while they do look nice, they don't give us a lot of like biomechanical interpretation. So I wanted to compare them to their biomechanical movement trajectories and show you what it really means when there is a high relevance placed on a certain trajectory. So on this slide here, we see a blown up version of the ankle inversion trajectory for cluster number 1 in universe, meaning that this is with respect to the universe shoe, the universe running condition. Again, all of these individuals that were assigned to this first cluster seem to have a high relevance placed on the ankle inversion. The graph here on the right represents the average movement trajectory of the ankle inversion for these 10 people that we looked at here. And the universe condition here expressed in blue seems to separate itself quite well from the other 3 movement trajectories. So it seems that all the models, all the artificial neural network models that were trained on subject-specific data learned to look at ankle inversion when the task was classify this movement pattern as the universe shoe. So there is a clear difference here. If we now look at a scenario where there was very little relevance placed on these ankle inversion trajectories, so this is now for cluster number 4, then we see that the biomechanical trajectories here on the right-hand side, they do not separate as well. So these neural networks, or for these participants, there was no value in looking at the ankle inversion because the movement trajectories would not separate themselves. So the response needed to happen somewhere else. Now, this concludes the 3 studies that I wanted to talk about today. In the first study, to bring that back into memory, in the first study, we looked at kinematic and kinetic variables that identified novice runners. And we learned that there are really certain aspects that we should focus on when our goal is to identify these novice runners. If we have a different goal, this technology or this analysis approach can help us to also answer that question. It can help us direct our focus. The second study took this a step forward, and we looked at generic and specific characteristics of elite runners. Now, a possible interpretation here is that generic variables, those that did not separate between our elite runners, could be those variables that we should look at when we are curious about generic elite running technique, something that all of the elite runners that we look at do. Specific variables, a possible interpretation, could be variables that characterize subject-specific movement strategies. So strategies that we should look at if we want to, let's say, design a runner-specific intervention program. In the third study we just talked about, we had the subject-specific adaptations to footwear, and this approach of matching artificial neural networks and layer-wise relevance propagation can help us to isolate subject-specific responses, and then we can use other analysis methods, such as cluster analyses, to essentially group those and find groups of individuals with similar adaptations. So overall, in all these studies, layer-wise relevance propagation helped us to direct our focus onto what really mattered for the artificial neural network. Oh, that was one too far. Now, before I let you go, I want to talk a little bit about the limitations that I've learned about while performing these analyses, and kind of raise a word of warning. First of all of the relevance scores that we've seen today and that I've ever analyzed are very specific to one model and one model only, meaning that we cannot necessarily generalize these insights to a model that is designed in like a different form. So different number of layers, different internal parameter structures, different activation functions, etc. So we really have to be careful if we want to make a generalized assumption as to these variables are always the ones we should focus on when we have a certain question. But I also think this is a good opportunity for research because the novelty of this method really lends itself to see if we can identify the effect that different model architectures might have on these relevance scores. Now, kind of in line with the first limitation here, these scores are really prone to model bias in that artificial neural networks are very sensitive. We can easily overtrain them, we can undertrain them so they don't generalize well, and we can also use biased data to train them so that the models learn something that we never really set them out to learn. Naturally, if we perform layer-wise relevance propagation on a biased model, we would also get a biased answer in that these variables are only relevant for the model, but the model is wrong, so the variables are wrong. So we really have to kind of know our limits here. We have to know how well this model works and what we could expect from this model before we do an interpretation or an analysis such as this. The third point that I want to raise is that negative scores are hard to interpret. Now, I have not talked about negative relevance scores today. I only shown you positive scores in that low scores represent variables of low relevance and high scores represent variables of high relevance. But negative scores are also an outcome of this analysis. And negative scores essentially represent variables that contradict the outcome of the model. So let's say we have a classification as in this movement pattern belongs to shoe A. Negative relevance scores would say, hey, hold on a minute. These variables here suggest that it's actually shoe A. And now we have this kind of like disconnect or this hard to interpret situation where we have these positive scores supporting the model's decision. And then we have these negative scores saying, hey, the other thing could be possible too. And it's unclear at this point how we can best interpret these and kind of draw the right insights from positive and negative scores at the same time. And the last limitation that I want to raise attention to or bring attention to is the fact that all of these analyses do not provide us a functional explanation for what's going on. Yes, we have learned that the model uses these variables for its interpretation or its results, but we don't know in the first place why these variables might look the way they do. Like, for instance, why is knee flexion high in this shoe or why is it low in the other shoe? We have no insights in regards to that question. The only thing we know is that from a data perspective, these variables have a lot of power, a lot of power to help the neural network find its solution. But we have absolutely no idea how that ties into the functional explanation of the system, of the human system. Now I want to bring this presentation kind of back to the beginning where I talked about the fact that our data sets are increasing in size and in complexity. And I think that this is not going to go away. In fact, with the advent of more variable technologies and the ease of data collection these days, it's highly likely that this number that I estimated in the beginning is a gross underestimate. So our data sets will become even larger and we will rely more and more on these data science methods, these artificial neural networks. And I think as a research community, we have to become comfortable with using these analysis procedures such as layer-wise relevance propagation to make sense of these data science models and really help us to redirect our focus because we're currently kind of lost in this jungle of variables and don't really know where to look. So I think or I hope that this presentation has given you a little bit of an idea of what is possible and a little bit of a, let's say, idea of what we can do to address this problem of big data. With that, I would like to acknowledge all the people that have helped me with the work. Obviously, there are more people than I can put on one slide, so I only put the main players on here. Thank you to all of them. And naturally, thank you to my funding agencies, Alberta Innovates, Biomechanic, and the University of Calgary. But most of all, I want to thank you for listening to me today. And if you have any questions, then I'm happy to answer them.
Thanks, Fabian. At this time, I'd kindly ask the audience members, if you're comfortable, to please turn on your Zoom camera to allow for a better discussion. To ask a question to Fabian, you can use the raise hand function under reactions on Zoom, or type your question in the chat box and I can read it aloud. I'll start with a question, Fabian. Your chart that showed your high relevance and low relevance variables across the stance phase, and or the stride cycle, you commented that there were low relevance variables near heel strike and toe off, which you interpreted to mean there was a generic strategy at foot contact and toe off. Is that correct?
So you're talking about the second heat map that I've shown, right? The one with the elite runners?
Yes.
Okay, just let me bring this back so everybody has it on mind. Share screen again. This one.
So yeah, and then you had those simple, just two lines, like a blue line and a purple line that each had two peaks.
I had, you're talking about this graph.
Yes.
Okay.
So I just find it surprising because I feel like there's a lot of interesting research that shows even extremely elite runners, when you capture them at the snapshot of foot contact, they actually have wildly different foot contact strategies. Mm-hmm. But then your results are kind of showing if there are low relevance variables, they have a fairly generic kinematic pattern.
Well, it's I mean, that's one interpretation, right? So I think it's all relative, right? For this study, we had 20 runners, and the goal was to see what separates best between these 20 runners. And I guess what these curves here show is that the early stance phase, the early stance phase, like these first 15-ish% here, if we look at the variables of low relevance, so the kind of left-hand side of the spectrum that I've shown, then these variables are predominantly derived from like the early stance phase. So this would suggest in a generalized pictures, right? Not in a very specific picture, that in this data set, our runners did not separate well when looking at this early stance phase. Now, I am not super into the literature, so I cannot tell you how many different papers are out there that say the early stance phase still has a lot of variability across runners, especially in elite runners. But I think I need to be careful here saying that that the early stance phase is often generic. I think that these insights only relate to the results that I've shown here today, only to the data that I've shown here today. And it might be that because we looked only at 20 runners, all male, all training to finish under 2 hours and 30 seconds, right? Like it could be that this very homogeneous subset of elite runners just didn't separate well in the early stance phase. There's still a possibility that if we had a bigger data set, if we had a less homogeneous elite running data set, that the early stance phase would become more important. So it kind of ties into this first limitation that I spoke about. These results are very model specific, right? We have to be very careful to generalize.
Great, thanks for that. Andy, please go ahead.
Hi, Fabian, thanks a lot. That was a great talk. if I heard you correctly, you mentioned in the first study and then again in the last study that you only used when the model was correctly able to identify individuals. You then use those correct classifications to develop the layer-wise scores. So if that's correct, I guess I have two questions. First one, just in general, how good were the models at this task? And then second, if you were to take examples where the model was not able to correctly classify these individuals, do you see this similar type of trends or patterns in these propagation, layer-wise propagation scores?
With respect to your first question, I actually have some graphs handy. So this graph here shows the accuracy of the model for the first study, right? The study with the novice runners shows the accuracy of the model with respect to the individual participants on the x-axis. And the color coding here represents the the three intervention groups that we had. And you can see that on average, the model's accuracy is somewhere in the high 90s. Obviously with some people in here where the model really kind of like dropped the ball. I have another graph kind of helping with that. I hope the next slide here shows up. So this is also for the first study where we compared the accuracy of the model with respect to how many variables we can use for the prediction. And here we can use, let's say, up to 1,200 variables. So this would be the entire data set, or up to, let's say, less than 200. So I think this was, the cutoff was 10 variables. And you can see that the best accuracy that we would ever get if we have all the variables, again, is somewhere in the high 90s. So for both of the studies, or for all three of these studies, the average accuracy was always very good, first of all. Now, the second question was with respect to the relevance scores of correctly classified strides or classified data patterns. There are two approaches basically to deriving relevance scores. So one approach could be that you, like we did, we only look at those relevance scores when the model is right, because we're only interested in what is going on when our model is well trained and can predict stuff well. Like we're only interested in working situations where the variables really predict the outcome of the model. If we have a negative or if we have a misclassification, then obviously the model didn't work well, and then why would we even consider the relevance scores? Because they might lead us to something that is not indicative of what we're looking for, right? There's a second approach where you just train the model as a whole, and you then look at a hypothetical classification. Because we go from left to right, right? We go from output to input. We can create a hypothetical scenario where we just look at a classification that was correct, and we see what the model would theoretically use for that. Now, this is a little bit less, say, data-driven in the sense that we don't look at one particular movement pattern that was correctly classified. We rather look at an overall model that was trained and see what it would use. I think, if I remember correctly, this was done by Fabian Hoitz, who presented, I think, 2 months ago here in the HPL seminar. He chose the second approach. And I had this conversation with him, and he said that the comparison across the two approaches is rather similar. So in both cases, you would get good results with respect to relevance scores. But I always found that the approach that we chose is really more with respect to the data itself and less with respect to like the hypothetical case of the neural network model. Does that answer the question, or was that way too far off?
Yeah, no, I think that answers it. So basically it's, yeah, obviously if a model doesn't work very well, then the layer-wise propagation scores are kind of irrelevant. So there's,
yeah, that's kind of what I'm thinking. So I didn't mention that in the presentation, I guess, but whenever we did train our models, we used a leave one out approach, right? So we would essentially say, here's the model for one subject. We have 40 steps total, train on 39, classify the 40th step. And if that is correct, we look at the relevance scores. If that is not correct, then neglect this and move on. Yeah. I wonder if Andy froze there.
I'll just wait one moment in case he has a follow-up. Okay. Great. I'll read a question from the chat from Dovin. I'm imagining that your inputs are absolute values or values normalized to max. If that's the case, are we biasing models to find differences where the value of the signal is largest?
Yeah, so the inputs are normalized. Let's say you have different movement trajectory in knee joint movement and let's say ankle joint movement. Obviously, the magnitudes are different in the raw values, but we would always normalize the trajectories to the range of -1 to 1 so that the model considers all trajectories to be equal in their contribution. So it can be that a small difference in, let's say, the ankle joint motion is equally well considered as a big difference in the knee joint motion, so to speak.
Great. Maybe I'll ask another question. A lot of your analyses were done on data sets that had relatively high sampling, and then you interpolate it to whatever percent stance or percent stride. And since you're headed to the wearable tech industry, I'm wondering if you can comment on how you think these methods apply to data streams that could be happening at a much lower sampling, like 1 Hertz data.
Yeah. Well, I think the biggest benefit of these methods is basically to tell us where to look, right? If we can kind of create a scenario where we test in the lab setting, we have a high sampling rate, and we then perform one of these analyses, you might learn that some random variable, let's call it variable X, really has predictive power for this question. So then when we then head into the field and we have a lower sampling rate to deal with, we can still try to kind of look at this one specific variable. And if we, or in the first case, the one specific movement trajectory, you know, if we already know that the ankle joint motion in the coronal, sorry, in this, what was it? Sorry, it was the hip joint motion in the coronal plane is very relevant. And we can look at that in the field, even though we have a lower sampling rate, but we can neglect the other two movement directions, for instance. So I think, again, the benefit of these methods is simply to help us kind of go through this jungle of variables and really like redirect our focus back to where we find the best answers according to these models.
Great. Benno, please go ahead.
Yeah, thank you, Fabian. You have shown that you have presented a method that allows you to be advised where to look. So what would be the next step in the analysis? Because at the moment, this is not a biomechanical question. This is a data question. How do we go back to the biomechanical question?
Well, I would argue that the biomechanics research in the past has focused on hypothesis testing, right? Like we design a hypothesis, let's say shoe A and shoe B vary in there or have different effects on the knee joint motion. And then we test the knee joint motion and perform a t-test, let's say, or an ANOVA test to compare the values and see if this hypothesis is correct. Now that we have a method to basically collect all the data that we want to collect and see which aspects of this data have a strong predictive power, which aspect of this data really can help us, we can now design hypotheses as to why these aspects of the data might be important and can go back to hypothesis testing. So I think it's a different approach to biomechanics in that we don't start off with a model hypothesis. Instead, we start off with a way where we collect all the data and then build the hypothesis in terms of like, why would this data vary at this point in time? And start to explain that with the biomechanical principles that we know.
But the biomechanical question is a mechanical or a biomechanical question.
Right.
While your approach is a statistical approach. Now, where do you get the actual thinking of a mechanical behavior back into the project?
Well, I think you, so to speak, never lose it to begin with, but I think the focus is more important now that we know where to look. Like, we now have, now that we have these statistical methods that tell us where to look, we now have to add our biomechanical knowledge and start thinking about, why would we expect to see a difference here? How can we explain this different now with the biomechanical principles that we know?
Thank you.
Welcome.
Philip, please go ahead.
Okay, so my question is related to some extent your initial example where you talked about, okay, when we see somebody in the distance, we can recognize them by their gait. I would in general also argue you could probably identify individuals that you know when they're just standing there. So now the question potentially is it actually kind of like individual movement patterns that arise from, let's say, potentially specific strategies within the nervous system? Or is it largely dominated by anatomical differences between individuals?
Well, in the case where you look at a person just standing there, I would see you only have largely anatomical differences to look at. So movement patterns don't play a role in that scenario, right? But I think that if you look at, if you take the visual example that I provided, let's say you see someone moving, you obviously have more data than these artificial neural networks models that we train, right? You have the visual data, you have the anatomical differences, and you have the movement strategies that you can compare with the information that you have stored in your brain, that you can compare with what you know. So I think that now, I think that there are anatomical differences that can help you with a recognition of friends and family and people you know. I think there are movement strategies that can help you with a recognition of friends and families and people that you know. And I think you can combine them both together to get an even more accurate recognition in that sense. So I don't think it's one or the other. I think it's a combination of the two. But in our approach, we have shown that looking only at the movement strategies, we can also do it.
Okay, and then just out of curiosity, do you know anything about potential like identical twin studies that look into this sort of kind of gait recognition?
No, I don't. I don't know of any identical twin studies. I know of studies in the psychology department that started to look into like moods and behaviors and see if you can recognize those as well. And it seems that happy people, sad people are all recognizable based on their movement patterns, basically. Yeah.
Interesting. Okay, cool. Thank you.
Yeah, you're welcome.
I'll circle us back to Dovin's question and read a follow-up from the chat. So his initial question was about if it's the absolute values are normalized to the max. For example, if a signal has a value of 5 at initial contact and a value of 100 at mid-stance, is it going to weight a 1 unit difference the same at each point? Using that -1 to 1, are we going to find relevance that is high when signal magnitude is high? In his example, a 20% change at initial contact and a 1% change at mid-stance are weighted the same because they have the same absolute value.
Let me double check if I understand this question. I'm going to read it real quick.
Yeah.
All right. So for the first part, do we what was the question? Do we find differences when the signal magnitude is high? Let me address it from this point of view. We've started looking at the effect that our normalization procedures might have on the outcome. You know, like Benno pointed out, technically this is a statistical method. So if your data varies, like if your magnitudes are different, then we can place a relevance there, because from a statistical point of view, the data points vary here, and therefore there will be relevance here. If we have an appropriate normalization procedure, so if we normalize within one subject, we normalize, so we can choose our normalization factor as one factor per subject, so that within the subject, the trajectories still vary, but they all are normalized to the same maximum. Or we can choose the normalization factor within one trajectory. So in each trajectory, we always have aspects from -1 to 1. And depending on which way we choose this normalization factor, we can somewhat influence where the relevance will be placed, right? If we normalize trajectory specific, we will always have a maximum value in this trajectory. And then it's more likely that we would find differences as to where this maximum occurs, opposed to the magnitude of this maximum. I hope this makes somewhat sense. So I guess just to summarize this, the normalization procedure has an influence.
I'm going to take a lull in my wife's simultaneous meeting to jump off the chat and actually just talk.
Yeah.
Thanks for bearing with my confusing chat.
No worries.
Basically, my question was just, is it possible that there's biomechanically relevant information when the signal magnitude is low? So maybe we're more constrained at that initial contact point, so all the values are lower, but our relevance scores are overlooking important differences there because the signal magnitude is lower. So the percentage difference is higher, but the absolute value, and therefore what we're considering relevant, is lower.
So I think that, yes, if you're, let's say if your percentage change, like even after a perfect normalization, let's say your percentage change in the early stance is smaller than your percentage change in the later stance, then naturally the neural network would learn to look for the bigger difference, because that better classifies between the conditions that you're looking for, right? You just have more, let's put it this way, you have more room for error where you have bigger magnitude differences after normalization, that is. Now, if your task is complicated enough that these bigger magnitudes themselves are not sufficient for an accurate model, then the model will then, well, I presume the model would then go ahead and also incorporate the lower percentage differences to make an accurate decision. So I think it's totally possible that we kind of get distracted by this and we look at the raw numbers over the biomechanical functional reason that might be important here. Yeah.
Osman, go ahead.
Hey, Fabian, thanks for the talk. I was wondering, how did you select this neural network model? If you change the architecture, like more layers, or deeper models, or any detail of the model, like activation function or whatever, would it show different results, like less relevant features became more relevant or such?
So there's actually quite some research, I think, on like slightly different, well, there's little research on slightly different architectures on the same data set, mostly done by the person who introduced this method. I think in his PhD dissertation, he wrote about that. In my personal experience, I played around with multiple number of layers, and I played around with different activation functions, like the ones I predominantly use is the hyperbolic tangent or the ReLU, rectified linear unit activation function, right? I didn't compare relevance patterns all too much across these different architectures. I only compared model accuracy across these different approaches. And I've always been a strong proponent of keeping things simple when I could. So if I found that one hidden layer was sufficient to reach 90% accuracy and a second hidden layer would only add, let's say, 1% accuracy, then why do go the extra mile, right? Make the whole thing a little bit less complex and make it a little bit easier to look at the interactions. I think there's also quite some research done on how many layers you would need or how many levels of abstraction you would need to get accurate results. And I think a strong comment is if you have a single hidden layer with twice your original input size, you already have a data space of such high dimensionality that your, let's say you have a binary classification problem, becomes really easy to solve for this model. So again, I would argue go back and stick with the simple model in that case. One point to this, I think we can look at solution spaces with respect to, let's say, artificial neural networks try to optimize for a minimum, right? So we can look at this solution space as some sort of like a hilly landscape with some local minima and maybe there's a global minimum. If I remember the research correctly, then these local minimums that can be found, so there can be different local minimums, different optimal solutions, are somewhat comparable in their model accuracy and also in their relevance scores. Yeah, but for this, I would recommend going back to the research group in Germany who introduced this. They did more work on this than I did.
Great. Wolfgang, please go ahead.
Yeah, first, thank you very much for this nice presentation. Because of the time delay, I don't know whether I'm still focused on all what you told to us. But related to Benno's question, did you ever do the same classification on the basis of the angular momentum? Because I think you just mapped some data, but drawing any consequences on where to look at mechanically doesn't make sense from an epistemological point of view.
I agree. To answer the question, no, we have not looked at angular momentum. I didn't have that data available.
Because I think this would be a way to go to biomechanics.
Mm-hmm.
Because when I saw one of your data set, especially with respect to the ground reaction forces, mediolateral forces, you saw all the time in all angles, you saw in all joints, you saw mediolateral abduction and adduction importance. And this could be, I would say, a latent variable behind all the things which is the same. And this would be a biomechanical question.
I agree. Yeah, like I said, one of the biggest limitations, especially for the work I've done, is that, you know, I stop at the point where I know where to look, but I don't really do the biomechanical interpretation at that point, or I don't use the right question there.
I think it's not where to look at. It's where to look in your model. But this is far from, I would say, reality. Because one of the major questions of biomechanics is which variables are controlled in which way and in which moment. And at the moment, I would say the model is a nice tool to map the data, but nothing more.
Yeah, I would agree. It's a very nice tool to map the data.
Yeah. Okay.
Yeah, it's a good point. Yeah, I should definitely. I'll think about that a little bit more.
I'll throw out one more question, Fabian. And again, this is focused on because I know you're going to a wearable device company. A lot of these companies spend a lot of effort in generalizability and like really minimizing false negatives and false positives. And I know all of your projects weren't necessarily binary classifications where you could have like false negative, false positive, but I'm just curious if you could do another study more focused on the generalizability of these approaches. And I know you already mentioned this is a limitation, is that these are for the models you developed. What would you do next to better understand generalizability of these approaches?
Well, personally, I would be really interested to kind of train a model, do the analysis, and then let this analysis inspire, let's say, a secondary analysis where we only pick those variables and we try to answer different hypotheses with these, right? It will probably, now I'm speculating here, it will probably turn out in a way where some of these variables are really good for the model to, let's say, predict a different running condition, but they are terrible for anything else. So I think, again, generalizing these results, so let's put it the other way around. If we want to do layer-wise relevance propagation and generalize these results to a broader spectrum, we would need a very solid neural network model that generalizes well to different data sets, if that makes any sense. So I would say if your goal is really to generalize the whole thing, we need a big data set of very different individuals, a lot of different individuals, and depending on the question, a lot of different conditions as well. Let's say you're looking at running shoes and their effect, you would need, I don't know, I'm spitballing a number here, let's say 40, 50 different shoe constructions. And the model needs to perform well on all of these situations to really be certain that the results that we get for this analysis are generalizable insights. Yeah, and I, like you always bring up the point that I had into the variable industry. I think there is two aspects to consider here. Like the one aspect is that this is a tool that can really help us to develop variable algorithms that, let's say, count steps or something like that, or that really look at one specific task where your goal is really only to take the data and get as much information off the data as you can. But it's not good to use to do like, it's not good to take this as a direct interpretation in terms of biomechanics. Hey, this is what leads to this, this outcome.
Great. Other questions from the audience? Wolfgang, please go ahead. Just don't forget to unmute yourself.
Sorry. One question with respect to the time normalization. When I understood it right, you normalized in one publication the data for the ground reaction force 60% and the swing phase was 40%.
We normalized from touchdown to touchdown of the same leg instead of from touchdown to toe off. So that meant that about 60% of this time normalization represents stance phase, while the last 40% represent the swing phase.
Okay, so if someone is limbing, it hasn't any influence on your normalization, no?
You mean as in like their step cycle is shorter, or what?
Yeah, not every step cycle has 60-40.
Right. That's true. I would argue in this case, we did this study on a very homogeneous data set, right? So these were all very strong runners. So I'm pretty sure that we have some differences in the length of their step cycle, but overall, I think none of them were limping.
Okay. We know that there are also different runners.
It's true.
Forefoot runners and rear foot runners, and with short contacts and more push phase, and yeah, okay. For me, it would be just interesting how big is the influence on this normalization.
Mm-hmm. I mean, I didn't compare that, so I can't really say that.
Yeah. Okay.
Yeah, but I would argue that, we saw in the first study where we had novice runners, we saw that the early stance phase, so the first 30-ish%, which close to heel strike were actually quite telling of the different runners. So there was a lot of information contained in this period. While in the second study, we saw that the early stance phase, even though it was only 60% of the overall area, right? The early stance phase was kind of like less helpful compared to the first study. So I think, first of all, the normalization factor will influence this. But second of all, also the population that you look at might have an influence on this.
Yeah, this question just came into my mind when the question arose with respect to generalizability, because you are actually constructing subjects which are very homogeneous, and then you want to generalize. I don't know if this step is correct.
Yeah, well.
No, I think in principle, these data cannot be generalized. It's an epistemological problem because of the holism problem of Duhem and Quine, which has been stated 150 years ago already, before Popper started.
Yeah.
We just have to stay with our data, and it's a good, I would say, evidence to go further, but not more than this.
Yeah, this is a point that we all seem to reiterate, right? Be careful with the generalization that comes from these models, for sure. Yeah, and another thing is, sorry if I bring this up again, but with the subject-specific models that we had in the third study, right? These were 36 subjects. We had 4 running conditions, and we can already see that we can basically isolate 4 different groups in these 4 runners. So I think subject-specific models might be the way to go in the future to look at like just one individual, right? We now have all the tools that we need to basically do these very, very specific analyses. So, yeah.
I'm smiling. This is a big surprise for me.
Any final questions from the audience? Okay, please join me in thanking Fabian for his presentation, and thank you to all the audience members for their questions. I hope everyone has a wonderful day, and I hope to see everyone again next week at the HPL seminar.